Thank you, Sousa. What an incredibly kind introduction. I have to have my mother stop writing them, though. It's really embarrassing. But I'd like to start out by saying I come to you this morning with a sense of urgency. The title is fairly benign, but the situation that we as a community find ourselves in could not be more urgent. So this is a shout out to Team Science. It's a different talk, but that's so relevant for this meeting. And what is unusual about this community, although not other rare disease communities, but it's very unusual for cancer researchers and cancer physicians to be as involved in the community of patients and families as Fanconi anemia requires. So teach us how to do this better. We need you and you need us. And I just want to shout out to my colleague, Dan Johnson, my research partner, who will speak later this morning on some of the actual science that we're doing. But today I want to talk to you a little bit about what I've learned. And maybe the most important disclosure is the last one in red. And that is, like most clinicians who have treated adults with cancer, I have no personal experience diagnosing or treating a patient with Fanconi anemia head and neck cancer. And that is true for most oncologists in the world. And that is simply the reality that we need to reckon with. So I had the privilege of speaking at this meeting in 2008. I was invited to join the Board of Scientific Advisors in 2019. And in my capacity on the board, I came to appreciate how urgent this crisis was in terms of the incidence and how little we knew about how to treat the patients with cancer. So we all know that once a patient survives their bone marrow challenges, they are at exceptionally high risk for solid tumors. For reasons that we do not understand, the incidence of squamous cell carcinoma is exceptionally high in this population. And squamous cell carcinomas of the head and neck are the most common malignancy that individuals with Fanconi anemia will develop, 500-fold increase. One of the most important points that I want to make today, and I hope we continue to discuss this, is most patients who are diagnosed with head and neck cancer will have no history of an antecedent premalignant lesion. We like to think that they do, and occasionally we will see premalignant lesions, but the vast majority of patients come to us with cancer. That is not true in the Fanconi anemia population. So we have an incredible opportunity to get together and start sharing our data, the clinical experiences, the response to treatment, the best approaches to monitoring, because these premalignant lesions in Fanconi anemia are a sign that something else is coming. We just don't know when. And in the ideal world, we would have a clinical trial platform that we could activate all over the world so that when a patient with Fanconi anemia develops a premalignant lesion, we can analyze it biologically, and we can then randomize that patient to a candidate chemoprevention strategy, because the best thing we could do would be to prevent these cancers from ever happening, because once they happen, they are really difficult to treat with systemic chemotherapy. So when I learned how to be a head and neck surgeon in the late 1980s, surgery was the primary treatment for head and neck cancer. Today, for Fanconi anemia patients and almost all oral cavity cancer patients, surgery is still the primary treatment modality, but surgery only works if we can get every single cell, so the margins have to be clear. We have to really look carefully for regional and distant metastases and think about how to manage them. The challenge for Fanconi anemia patients is that the most effective chemotherapy agent, which is cisplatin, which was FDA approved in the 70s, is intolerable. You heard yesterday about Nigel's daughter's experience being diagnosed with Fanconi anemia when she had a catastrophic toxic reaction to platinum chemotherapy. That is still sometimes how Fanconi anemia patients are diagnosed in adulthood. Over the years, radiation oncologists have worked very carefully to show that we can deliver external beam radiation therapy, whether they be protons or other forms of therapy. But once again, this has not been carefully studied. There's certainly been no clinical trial that I'm aware of. And as I mentioned in my disclosures, there are very few centers or clinicians with a substantial experience of Fanconi anemia-related cancers. And there's a very limited literature, especially large clinical series or any interventional clinical trial data on treatment, the toxicity of the treatment, and the survival outcomes of the patients. So when I say head and neck cancer, what I mean is oral cavity, oropharynx, and larynx. This is a sagittal MRI scan. The vast majority of Fanconi anemia patients will have oral cavity cancer. I remember, actually, Alan DeAndre and I were at a meeting together many years ago when we discovered that HPV, or human papillomavirus, was a common cause, or an increasingly common cause of head and neck cancer. And that is true, but not in Fanconi anemia. So at the time, we hypothesized that that was the reason why the Fanconi anemia population was getting head and neck cancer, because these were younger people who didn't have the traditional risk factors of tobacco and alcohol exposure. It must be HPV. Well, it is not. So I don't want to spend more time on this slide except to say that this slide also shows how hard it is for an inexperienced clinician to examine the patient. We, as otolaryngologists, are experts at examining the oral cavity, the pharynx, and the larynx, and we need to be involved, and we need to understand that we have to do very careful physical exams. Dentists are also very good at detecting oral cavity lesions, but it is difficult for non-dentists, otolaryngologists who don't have the equipment or the experience to look in the oral cavity, and to see the hypopharynx and larynx requires specialized equipment and training. Even for sporadic head and neck cancer, there are very few tools in the clinical toolkit for systemic chemotherapy. As noted, cisplatin is by far the most effective tool. This is not one that can be used in the setting of Fanconi anemia. In 2006, the first and only molecular targeted agent was approved for use in sporadic head and neck cancer. That is the EGFR monoclonal antibody cetuximab. Our next speaker will talk a little bit about EGFR as a potential therapeutic target in Fanconi anemia. Docetaxel is a chemotherapy agent that appears to be tolerated in most patients with Fanconi anemia, but again, I am playing with a very limited deck of cards. I only have sporadic case reports, and those are incomplete in terms of toxicity and outcomes. So once again, we need to think carefully about the systemic agents that could be tolerated, ideally identify predictive biomarkers so we can match the right patient with the right treatment and treat everybody with Fanconi anemia cancer on a clinical protocol so that we can learn and do better for that patient and for the rest of the community. In 2016, it was a banner year for head and neck cancer. The immune checkpoint inhibitors, pembrolizumab and nivolumab, were FDA approved. There has been a cautionary concern about the dogma in medical oncology is that a patient with a prior bone marrow transplant cannot get an immune checkpoint inhibitor. There are some case reports showing in Fanconi anemia head and neck cancer that it is tolerated, but we have no data that it is efficacious. So once again, we need to test, we need to iterate, we need to understand. So motivated by the paucity of evidence, and around this time I joined the board, we started the virtual tumor board experience because there was such a great need on the part of patients and treating physicians to understand more. And so a very talented medical student, now an ENT resident at UCSF, Rex Lee, did a literature review, and we published this in Clinical Cancer Research in 2021, so about a year and a half ago. And at that point, there were 131 cases of squamous cell carcinoma. Most were the head and neck, but there are 13 esophageal squamous cell carcinomas. And I firmly believe that the old-fashioned way of treating cancer, which is by organ site, makes no sense in terms of biology, and mucosal squamous cell carcinoma in a Fanconi anemia patient is probably the same disease. So it deserves being looked at together. So most of them if they were cured, it was with surgical resection with curative intent. That is the primary treatment modality. Radiation, as I mentioned, was tolerated in most cases, although there were treatment breaks necessary. DNA-damaging chemotherapy is not tolerated. And EGFR targeting agents, and the only ones that were reported were cetuximab, were tolerated with some reports of efficacy, but again, the data are tragically incomplete. Limited reports on immunotherapy. So my message is we need more evidence. We need more data. We need to decide what are the critical common data elements: clinical, pathologic, treatment, toxicity, and outcome. And we need to work together all over the world. There's no tolerance for separate platforms, for siloed approaches, because the disease is too rare and the need of the patients is too urgent. So this is a table from the paper just showing you a summary of the clinical and pathologic characteristics of the patient. I won't go into the details. You can look at the paper. And at the end of the day, the recommendations are just as I have illustrated again and again, and I'm embarrassed to say that these are the same recommendations I would have given had I given this talk 30 years ago. And there's something deeply wrong with that, because I know a lot about squamous cell carcinoma of the head and neck biology now, and I would do it differently for that population, but I don't know enough about the biology of squamous cell carcinoma in Fanconi anemia patients. We don't know enough, and we have to learn more so we can do better. So, in summary, the key gaps in our ability to prevent and treat cancers in Fanconi anemia patients is we have a paucity of models. You'll hear at this meeting today about the models that we are all trying to develop. We have to share these models. We have to critique each other's models. We have to validate findings in one lab, in another lab. This is so urgent. And that includes cell lines, patient-derived xenografts, and genetically engineered mouse models. Again, we have an embarrassingly limited amount of information. And this is hard information to get, because if you go to an electronic medical record, all of the critical information is going to be in the physician or the nurse's notes. And so it's only going to be as complete as that physician and that nurse reports, which means we have to think about every single patient as an N of 1 clinical trial. We need to have Fanconi anemia case report forms. We need to tell the treating clinicians what information needs to be inputted. And until we do this, we're still going to be stuck with this morass of incomplete information. So we've been doing this Fanconi anemia cancer virtual tumor board now for several years. I have learned so much, but the most important point I've learned is it's a need from patients all over the world. I'd say half of our requests right now are from patients outside the United States. Number two, we have never met a referring physician who had ever seen a patient before with Fanconi anemia-related cancer, and this is never going to change. This is just a reality. And number three, while those of us who participate, the clinicians, the surgeons, the radiation oncologists, the medical oncologists, are really learning from talking to each other. We have a harmonized voice. We know what to listen for. We know what to suggest. We don't know anything more about the disease than we did when we started 3 years ago. We make recommendations. We have no idea if the recommendations are followed. We don't know what the toxicity of the treatments are that the patients receive, and we don't know what their survival is. So we have many, many critical gaps. And we don't know the safety and efficacy of immunotherapy. There are few clinical trials. You'll hear about one being developed in Europe, but I would argue that we need an N of 1 clinical trial model based on a deep appreciation of biology. And I personally feel like every cancer patient in the world should be treated on a clinical protocol, because patients on clinical protocols do better because the treatment they're receiving has been very well justified in terms of preclinical models, and people are paying attention to them, and the results get reported, and we get to learn in an iterative fashion. So I think we have a question. We don't really know the relevance of findings in one cancer to another cancer. I mentioned my own bias, which is that squamous cell carcinoma of the pharynx and the esophagus are the same disease. They're just a few millimeters apart. But in the world today, it's the thoracic surgeons who manage esophagus cancer and the head and neck surgeons who manage head and neck cancer. And we don't talk to each other. We don't share clinical trials. But molecular tumor boards have taught us that we need to do this, and that's another really big gap I want to point out. We do not have somatic sequencing of all tumors that arise in patients with Fanconi anemia. This is unconscionable to me. This technology is widely available, it's relatively inexpensive, and at least if we profile biologically all of these tumors, we would at least have some data that we could go back to our preclinical models and test candidate treatment and prevention agents. So I know that I'm out of time, and I would just like to thank everybody for listening today, and I hope you share my sense of urgency, and I look forward to further conversations. So thank you very much.